Thank you, Giovanni, and before you are leaving, because I know you have a busy day, I just want to say how much I appreciate being here for the very first summer school. So you should know you are really pioneers here. You are at the first school, and it's all due to Giovanni, who have actually come up with not just the idea, but actually also done all the work to have this first class. So I'm really hope, and I'm really very positive that it will be a success, and that this will only be the first in a series. And maybe, as you see now, Giovanni is the chairman of the European Academy, but that we also maybe in the future can expand and have this on a more global theater. So thank you, Giovanni, for inviting me here, and in particularly for organizing this. So I got this topic, and I have never actually given this kind of talk and never had this kind of course. So this is also kind of pioneering. So having this, it's a very broad, you can do whatever you want actually. And then eventually I realized that we will share the day here with Juliet. And Juliet, maybe you should stand up so they can see you already now. And maybe you can also give a short presentation now. You will come back later in the afternoon, who you are.
I have quite a loud voice anyway. My research is in severe and profound intellectual disability, and I have a real interest in qualitative research, and that's what I'm going to talk to you about this afternoon.
Yeah, thank you, and I'm very glad you are here. So when we got this mission, I should focus on quantitative research methods, and Juliet will then focus on qualitative. So therefore, that I don't have them here is not because I think they are not required, not good. It's just that I know that the entire afternoon the focus will be much more on qualitative. So for me, it's no really difference actually between quantitative and qualitative. You should just use the method which can help you to answer your question. Okay. So then I thought I started with this picture. So this is different categories of research when we talk about the area where we are in disability, children with disability. One thing more, I will do PDF of this series and send to you. So you will have all figures. You don't need to do figures so on. You will have it. So you could see it as a link, which can all be overruled by what we call translational research, starting with basic science, coming into clinical research, doing epidemiology and health service research, and eventually, if there should be any meaning at all, it has to be implemented. And that by itself is a research area. How can I implement now what we have known? So the definitions you have here, so translational research, that is you are translating basic and clinical research into clinical practice. So that's actually describing the whole chain. The basic science aims to understand the basic structure and function of the nervous system. Clinical research, that is research studies on people, usually different people with different kinds of health conditions. Either you just describe them, you follow them, or you interact. Somehow you intervene, you give them an intervention, and you try to find out, did this help or not? So, and I would say when studying your research proposals, I would put most of you into this category, the clinical research. We have then the epidemiology. That's the study of the distribution and determinants of health-related conditions. There was actually only one of these, and I actually only found one what I would call basic science in your examples. You then have health service and social science research, and that means that you study social factors, financing systems, and other kind of processes, and you see how they affect the access. So how can our patients, how can they access, what real service do they get? And finally, we have then the implementation research, where we try really to improve programs and policies. So take 30 seconds. What kind of research are you doing? Where would you put yourself in here? So when I looked on it, I actually now, and there is no really scoring priority, I just took 4 examples from your submissions, which we will discuss then later this afternoon. And you see I took one from basic science, or at least partly basic science, one clinical research, one epidemiology, and also one on health service and speech. So we will have 4 different categories we will discuss later this afternoon. So before that, and during two lectures now, I will give you examples, and since the research I know the best is my own, I will of course use my own research as examples. So I will give you 3 examples here, and all of them are actually into translational research. The first will be what we could call proof of principles. It's basic science, because if you can prove that this mechanism or this effect is there, it can have enormous impact later on, if you remember now the chain. If you can show basic science, you can then move it. Then I will have one epidemiology, then how many children with developmental disability and cerebral palsy are living in the world. That's a quite small question, isn't it? And you will find out, you will see, when I started this, I got so actually surprised to see the status on this question. And the second one is how to develop an intervention for children with, and I have chosen then a project we are just starting with cerebral palsy in a low resource setting. So how should I choose then among my different research projects? And then of course I wanted to show then the one with the most impact. And how do you measure impact? I think it was very late in my career I started to understand that most of the research done does not have any impact because it's never cited, or it's cited maybe 5-10 times by the authors, but none. So if you should see really that a research project has an impact, you go and see how often is it cited by others all around the world. And the more traditional way is to go to the Web of Science, because there you can find out the different papers and how often they have been referred to. But you have also a more similar and wide one, the Google Scholar, because that you don't need any licenses or anything. And the Google Scholar is also much more liberal. They actually include all kind of references, while the Web of Science is more strict. It only take up indexed scientific journals. So it's more the old core kind of research, while Google Scholar is more. There are also different kind of research platforms, but I have not so much used them actually in this. So there you go to Web of Science and you just punch in either the article or the topic or the author if you want. And if you look on the authors, you can also find out how much has this author been cited. That's a way to see how much have been. And for me then, what I did, I have actually a few papers that have been really making enormous impact. And the first one is then on the normal gut microbiota and how that influences the brain. And you see that has almost been cited 1,000 times at the Web of Science. So that is impact, you could say. And the other example is a bit older, where we were training working memory in children with ADHD. And you see that has also been almost 1,000 times cited. Then if you just compare with the Google Scholar, you will see they have much higher citations. So I choose this then as good examples of research that has made an impact in the way that it has been cited. So the first thing you have to do is to really formulate a very strict research question. And actually, when I have seen your questions, in general, I would say they are rather diffuse and descriptive. So the first thing you need to do is you should have a very sharp research question. And actually formulating the research question, that's one of the really most important things. Because if you have a good research question, then the rest is following. If you have kind of a diffuse research question, the rest will also be diffuse, unless you are very, very lucky. So in this case, my research question was, can the gut microbiota influence brain development and have an impact on neurodevelopmental disorders? That's a rather precise question. So first you have to do literature research. So why did I actually got this idea? First of all, I need to know about the microbiome. That's kind of a general understanding. I need to know how can the microbiome affect body systems. Are there any human studies, or are these only experimental studies? I also have to know now, since I'm interested to see if the microbiome can affect brain development, I need to know the brain development and the mechanism. And then I'm coming now to the last question. Are there any other studies on the effect of the microbiome on the brain? Because that is my study. And then I could only, in the whole literature in the world at that time, and this was 2009, there were only one study on this. And it was actually published several years earlier on, where they could show that if you had this kind of a germ-free mice, you had actually programmed changes in the HPA axis, you know, the hypophysis pituitary adrenal system, so they could see changes they were. So there were only one study at that time. The microbiome, you will hear more about it this afternoon, so therefore I also thought this was a good example. So the microbiome, that is that how many genes do you have in your body? Anyone knows how many genes do you have, a human have in their body? You can actually almost see it here. So how many genes do you have? Nobody knows how many genes there is in the human genome. You can read, there are between 25 to 30,000 genes in the human genome. But at the same time, you have about 1 to 2 kilos of bacteria, viruses, fungi in your intestine and on your body. And if you look on those genes, there are trillions, or millions anyway. So that means there are a lot of genes in your body which is not your own. You are carrying around them all the time. And what you call now is the super organ, because if you count now those genes and your genes, you will find that this microbiome, these genes, they produce proteins, and these proteins can affect your body. If they are coming in, if they are translocated into your blood, these proteins can now affect your body. And this is actually the and if you read now, there is so much. This is one of the hottest research areas. So it has been from the millennium. So as you see here, the microbiome, all these bacteria in the gut, they can prevent invasion of pathogenic microorganisms. They contribute to the fermentation. They produce vitamins. They are involved in the metabolism of fatty acids and carbohydrates. They promote absorption of minerals, and they can regulate inflammatory response. And in particular, the later, that's a system response. It's not just in the gut, it's actually the whole body. And it has also been shown that if your microbiome is offset, you can have a lot of metabolic problems like obesity, the metabolic syndrome, diabetes, infections. So that is correlated to your microbiome. So then the question is, can it affect the brain development? So what do we know about brain development? This is kind of 1 week or 2 week or 1 semester course in one slide. So we know that the brain is controlled by a genetic blueprint. That's to the left, the genes. The genes are controlling the brain development. But we also know that there are a lot of environmental factors that can influence this blueprinted brain development. We also know that there are quite a lot of gene abnormalities, like chromosome abnormalities, copy number variation, single polymorphism, which gives you risk genes that will enhance the risk that you develop, for example, ADHD or autism. ADHD, we know that about 75% of the factors are hereditary. That means that it's very much genetic disorders. We know that autism, about 50% of the etiology are genetic factors. And even for cerebral palsy, there are emerging evidence that about 30% is genetic. So that's really shows that a lot of the abnormal development of the brain leading to neurodevelopmental disorders, they are due to these gene abnormalities. We have to know that. We also have to know on the right side that there are a lot of environmental risk factors. Intrauterine conditions, preterm birth, we know is in particular extremely preterm birth. More or less every child below 25 gestational weeks has some kind of cognitive aberration. And we have, of course, a lot of postnatal and childhood. So if we are then unlucky, or what we should say, or in a variation here, we have a lot of what we call health conditions leading to different kind of neurodevelopment. So that was a brief summary of brain development in maybe 1 or 2 minutes. So now the question. In this machinery, gene-environment interaction, can the microbiome, which I have given also, that's also 1 semester, 2 semester courses, brief background. So why do we expect it? Yeah, because if you look, for example, on autism spectrum, you will find it's very prevalent with gastrointestinal syndromes. More than half of children within the autism spectrum will have gastrointestinal symptoms, diarrhea or constipation, when they do tests, they often see that they have a leaky intestine. That means that the membranes are not really that tight. We also know that there is a correlation between gastrointestinal system and severity of autism. We know that this group of children, they have used and they are using excessive use of oral antibiotics. They eat much more of the antibiotics than other kids. Many have tried different kind of diets and probiotics, and there are studies showing that there might be some improvement. When they have been looking at the microflora in the feces, they have found that in many, many cases there is an alteration. However, they have not found a typical alteration, but there are often alterations. And we also know that autism spectrum, they have a lot of immune problems. All these suggest that there might be a connection. So what we did now, very quickly, we used a facility which is called germ-free mice. That means these are mice that are born by sterile mothers in a sterile condition. they get sterile food to eat. There are filters in the air, so there can come no viruses, so they are completely sterile. And this now gives us a good chance to see, is there a difference if you are growing up with or without a microbiome? So if we now see a difference between these germ-free mice and these mice with microbiota, we can say, okay, this is probably caused, we cannot be 100%, by this difference. So that is very simple. Of course, this facility you cannot find anywhere, and you must have kind of resources in order to use them. So first of all, we want to see, do they behave differently now, these germ-free mice? So what we can do, we can study motor activity in an open field test. We can study anxiety, and that's an elevated plus maze or dark and light. So the open field, we just study how much is a mice moving. In the elevated plus maze, there is one closed arm, that's the one vertical, and it's one open arm. If they are anxious, they stay in the closed. If they are less anxious, they walk on the open. The dark and light, then if they are anxious, they stay in the dark. If they are not, they go out in the light. And then we have the social interaction. I'm not going into that one because it's kind of difficult and take time. So you have to trust me there. So here you can see now the motor activity on the top. Let's see now, on the top. There, you see? This on the top, you see the first 2 times, there is, the first 10 minutes, there are not any difference. They are all active. So the red one, that's when they are running. The blue ones is when they are staying up, rearing, it's called. But already after 30 to 40 minutes, you can start to see a difference, and very clearly 50 to 60 minutes. So you see, while a normal mice stop running around, exploring, sitting in a corner, the germ-free, they continue. And to the right, you can see the statistics. You see the black are the germ-free, and you can see they have much more motor activity. So here we can say they have hyperactivity. They have a motor hyperactivity. Do you agree? And can you follow this? It's quite simple, actually, if you just have the right method. This one is a bit more difficult. And let's see if we can look at these. So here you have the normal mice walking. Can you see? It's walking, and you see it's mainly staying inside the closed arms. Can you see that? It's just poking out, but then continuing there. Look now, compare that to this one. This is a germ-free. There it's coming up. Still in the closed arm, rearing a bit. Let's see, poking out. Oh, look there, it's going out. And you know, it's 1.5 meters above ground. So you have to be quite brave to be out there. So do you understand? So this is just measuring now how much are they in the open arm and in the closed arm. If you have several, you can eventually find out this is a clear difference between these groups. So let's see what we found. So in the first one, you can see that the white one, that's the normal, and these are in the center, that's the closed arms. They are less the GF, while if you look on the open arms, you can see that they are much more there. So by this we can tell these mice are not as anxious. They are much more brave running out there. And then we have the social. I just give you the principles. So in the middle you see the chamber, and then you have another mouse, and then you have an object. And now you can see, is the mouse more interested in the other mouse or in the object? Because if you are socializing, you go to the other mouse. And you can also have an old mouse and a new mouse, and you can see how they explore. I'm not going into details because that would take time, but anyway, you can just see that there is a difference now in the social interaction. Okay, do you agree? Now we have showed that the microbiome, probably, because that's the difference, can change the behavior. So behavior is at the very end of what we are doing. Secondly, now we go and look on the genes. Can the microbiome influence the genes? So this is a collaboration. At that time, it was not so common with whole genome mapping. Nowadays, it's done many places. So we had a collaboration with Singapore, and this is what's called a heat map. So you see the green, that's when the genes are more expressed in the SPF compared to the red, the GF. And so you see there are many, many places where they are more expressed. At the bottom you see a few where they are less. And you also have three different cards. So in general we could say that if you are invaded by the microbiome, that upregulates several of your genes, which is not upregulated if you don't have them. Do you follow me here? And to the lower right, you can see now that there are 4 different pathways where you have the influence. So now we are starting to get the machinery together. The next thing we wanted to do, that is on the protein and the messenger RNA level. I mean, you have the genes, you have the behavior. Now we want to see, can we really find that these genes are upregulated? So this is nerve growth factor, that's NGF. It's BDNF, that's another brain-derived nerve growth factor. DRD1 is a dopamine receptor D1. So here we can now specifically look on these messenger RNA to see, are they increased? And just by looking, we don't go into details here, but now just look at the NGF. So this is a normal, and you see the yellow, that means that this mRNA is upregulated. You look exactly at the same for a germ-free. Do you see any difference? Very clearly, they are upregulated. And to the right, you can now see the different statistical means. So that means we can show now also that many of these messenger RNA is upregulated. So this now puts it all together. So by doing this basic science, we have for the second time maybe, because there was one other paper, we have now shown to the left that there are behavioral reprogramming. In the middle we have shown, and I haven't shown you everything, that the brain structure and function can be influenced. And to the very right, we can actually see that the genes have been up or down regulated. Of course, we have not really shown that these are connected, that they are causative, but of course this is what we assume, but there is a lot of more studies doing, because what we think is that the genes to the right, that they are upregulated by the microbiome, that changes the brain structure and function, which now changes the behavior. And if that's correct, this could actually now be a mechanism, for example, for the autism spectrum disorder or any other. Now just to show you, this was then the second and the first one really showing the behavioral differences by the microbiome. Within 1 year after our study, there were 5, 6 other coming. So it was really clear, if we hadn't published this first, somebody else would. And actually we lost half a year by trying to publish in Nature, but they did not believe several of them. So it was kind of lucky still that we got it out first. Now, of course, and I will not go into this, now after that we have tried to understand what are the mechanisms. So these are many pathways from the gut to the brain, And one is then nerves. There are both afferent and efferent nerves. So there is a neural pathway between gut and brain. There are different kind of endocrine cells sending out different hormones from the gut, which can pass up to the brain. There are also different inflammatory molecules, cytokines, interleukins, which can be transported up. And finally, there are bacterial metabolites. And there we continued actually and did an experiment, what is called a peptidoglycan, which is then in the walls. And by doing a lot of experiments then, we could actually show also for the first time now, and that is a recent publication, that this bacteria peptidoglycan actually can do these changes. So this is now proof of principles. And if you read, this one was just published, I wrote in the Developmental Medicine Neurology, just a very brief review about this, the background of this. So here you can find the whole story, and there also now the connection with the neurodevelopmental disorders, which probably we can discuss more at the very end of this day. So that was my first example. So maybe I can just stop here before 10 minutes, I take the second example. So do you have any questions here or any comments? Yep.
I'm just thinking of, because that's what you normally, the transference from animal studies to human studies are of course a vital part also, because the humans are not rats, so there can be differences, of course. So I'm just curious, so how far have the research come with starting to see the mechanisms in humans?
How far? I would say it's one of the most active areas. So I would say every week there are several studies coming in this area. I would say that we are still in the beginning. There are several examples where we indirectly can show that these mechanisms are going on also in humans. So I took them away, but for example, there have been studies where they have seen when you are taking antibiotics, when the pregnant mother is doing that, and the child during the first year. And they have been, in huge studies, been able to show that these children have another phenotype. They are more, what you could say, ADHD-like. They are hyperactive LS. So somehow you could say there is now an association between these. Also, they have done transplants of the microbiome in children with autism spectrum, which means that you first wipe out their microbiota by strong antibiotics, and then you infuse, they eat then the bacteria from what you call normal bacteria. And there are studies now showing that you can improve behavior and you improve gastrointestinal symptoms. Finally, there are now studies from children who are in low resource settings that they have another microbiota when they got this kwashiorkor and so on, and that this microbiota contributes to their development. So it's a very good question, but there is not a final answer. So as I said, so there are every week there are actually new studies coming in. But I think this is such a basic mechanism. It's such a basic mechanism. So I would be extremely surprised if you will not eventually find much more direct evidence.
I just often see when I read papers about animal studies and then the effect on humans, that sometimes it's not really clear if it is actually animal studies only or if there have been studies in humans too. And I think it's I think it's important when you read animal studies to sort of think that, yeah, this is animal studies and this is a good starting point. But we need to know that humans are not rats.
No, absolutely. And I would say if you are dealing with many other body systems, like the heart, like the kidney, there it's much more similar between animal and because the basic mechanisms are the same. If you come into motor systems, it's also still a lot, but as soon as you come into cognitive functions, what we are really dealing with, it's so big difference between the brain in a mouse and a human. So of course, you cannot just directly translate. That's the reason, if you remember, basic science was to the right, and then there is a lot of steps until we really can show that this works in clinical practice. Okay, another question?
Are there any study about epigenetic mechanism and microbiome associated with neurodevelopmental disorder?
In humans?
In humans, yeah.
Not what I'm aware of, but I'm sure there are.
Okay.
I'm sure there are, because that's another field which is very, very much. So I would think in the experimental animal basic science that they are studying these mechanisms as well. But in a way, I would think that the microbiome, that what the effect is probably through epigenetics. Because what's happening is there is a very, very early impact, which is that lifelong. So probably what's happening is that you have some kind of epigenetic mechanism there. We don't know how though, but that it's there for sure. So we could say this is probably an example of an epigenetic thing happening.
Thank you.
Okay, let me take the second example from this basic science, because then we have to hurry up a bit. So this is much more human now, because still you can make basic science on humans, which can be useful. And this has actually been extremely useful and is now used in clinical practice. But it all started then and this is in the 90s when we were studying children with attentional deficit hyperactivity disorder. And at that time, this cognitive functioning was not so much in focus. But we were really trying to understand what are the functional limitations in these children. And at that time, it said it was executive functions, it was state regulation, and also the processing of different motor and sensory information. So then we go down, executive functions. So these are old slides actually, but I think they are still valid. So you could say the core functions is cognitive flexibility. Now we have a psychologist here, so I have to be very careful. It's self-control, that means really, which is really one of the really core problems for children with ADHD. And it's also the working memory. But the working memory at that time was not in focus, the other two very much. And actually, if you do it more complex, it's problem solving, reasoning, planning. So this is now the cognitive part, not really all the behavior part. And we also at that time knew quite well that you have the medial frontal systems, really where you have the reward system, while the dorsolateral system were more the analytical and the executive problems. So working memory then, that's the ability to keep information online for a brief period of time. It has a limited storage capacity, so each of you probably would have different capacity to remember for a short time. And it was also the storage on preference. So we know this was an active process because there had been a lot of primate studies. So the working memory was depending on an active neural activity. These are examples. Can you, 852, can you remember that? If I give you my phone number, 4608755768. Can you remember that? Probably you can when you are dialing and talking with me. When putting it down, it's all gone. So it's a good example. But it can also be visual. Can you remember these spatial location? If you have the mathematics, can you keep 73 minus 7? You have to do that, keep that in mind, put +2, and then you get it. So in all these cases, you need your working memory, and this is another one. So this is working memory. So at that time, this is kind of fascinating, because it said working memory is fixed, it's a core function, it cannot be altered. And when we now later showed this, we got our manuscript refused several times because they said, this is not possible. You have, so at that time, and I don't know, Juliet, if you remember that, but at this time, working memory, that's something you are born with, you have it. You can't do much about it. So our research training question was, can training of working memory improve working memory? That's a very easy one. If it does, can we use this now to improve working memory in children with ADHD? And if that is possible, what are the mechanisms? Very easy questions. So the first, you see here we used a visuospatial working memory test. You see you have signs to the left, down, blue, 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 blue. And now they had to repeat that in the MRI scanner. And we could then do an fMRI testing or seeing where are the neurons active in the brain. And here you can see to the right, you have then the network, the active neural network of the brain when you are doing working memory tasks in the MRI scanner. This is so common nowadays, but this was actually in the 90s when it was very, very, very uncommon actually with fMRI studies. Okay, so now the question, if we now train children 40 minutes per day with a working memory program, and of course we had to do it in a game, so they were sitting in front of their computers. So they did this 5 days a week for 5 weeks. So 40 minutes, 5 days a week for 5. And then first you see to the down there, you can see this is the working memory. So you can clearly see a learning curve. There were enormous increase in their working memory capacity. So that was actually the first. They could actually improve their working memory by training. Then we did this on several groups of children with ADHD. And you see the learning curves to the right for different studies. And we could see that not in all children, and this is important, if you took an average, a mean, you could see you have an improvement. But some did not respond, while others responded very well. So in this case, if we took the 80% who really responded, they had a very good increase in working memory. They increased their attention. They had a general, when you did the Ravens, they had a general IQ increase, and they had also reduced symptoms. So these are the first two studies, 2002, 2005. And after that, there are probably 100 studies repeating this, probably 100 in different ways. But these were actually the first time these were shown. The second thing we wanted to know, the mechanism, is the dopamine involved? And as you know, most kids with ADHD, they get amphetamine or some other central stimulants, which is then increasing the dopamine tone in the brain. And similarly there, we knew then that 60 to 80% responded. And also if you gave this to students, ordinary people without, they also improved their working memory. We knew from the molecular genetics, where you looked at risk genes, that many of children had risk genes for dopamine transporter, dopamine D4 receptor, COMT, which is an enzyme breaking down dopamine, that they had anomalies in these genes. And finally, there were a lot now of coming to the mice models again, knockout where you had it. So we then use what is called a PET, a positron emission tomography. which means we can look, in this case, we then looked on the dopamine D1 receptors, which is part of the dopamine system. We couldn't do this on children with ADHD, so we did it now on healthy students like you, 10 to 28 years old, maybe a bit younger. They had 25 days of working memory training the same. We did PET before and fMRI and cognitive tests before and after. And these are the results. These are the cognitive tests we did, and we could show that all of them except the Ravens actually improved. So these are students at the Karolinska Institute, no ADHD symptoms. They get a working memory. They improve their capacity. Secondly, we took an fMRI before and an fMRI after, and this is a difference. So you can see in this group, you had an increase in the bold signal, which means there is an increase in the neural activity. The only difference was now that they have been training for 28 days. And you could see a change in their brain activity. Secondly, we now so these are the bold responses. So bold, I can't go into details, but you see the blue. Here you see before and after. So you see in many, many areas of this neural network, you had an increase in the bold, telling us there is increased activity in this part of the brain. And finally, we did the PET. And here we look on the binding potential. But then you see, to our disappointment, we did not find any correlation. So that was kind of really sad. But now, and this is important for you, you cannot only think linear. So do you know what I mean with think linear? Because in most of our brain, at least you are all an association, you're only thinking you have the x-axis, y-axis. If there is a correlation, it is like this. It's linear. Do you understand what I mean? So what we then looked through the literature again, and this is now, since we talked about how much can you translate to humans from monkeys, this is actually made in primates. So on this slide, you have the cortical dopamine levels here, and here you have the activity, the strength of memory fields, working memory performance in monkeys. And what you clearly see here, that if you have low cortical dopamine levels, you are not as good as if you have higher. But if you have too high, you have a reduction. So this is what is called an inverted U-shape. So thinking about this, could you expect to have a linear association? Could you expect that? So therefore we now used an algorithm where we were looking for different kind of non-linear. And then you see, when we did that, you see the brain areas. You can see on the, let's see if I get this. You have 5 different brain areas, and for each of them you have the plot of this binomial correlation. And then we got significance in 4 of these areas. And that, as you see, we could actually publish this in Science, which is not quite different. So this was actually the first time anyone had shown that by doing an intervention or a training, you could actually change the molecular mechanism in the brain. So the answer here, if I put it all together, is that attention deficit hyperactivity disorder, it's you have a lot of behavioral symptoms, you have impaired cognitive functions, and that's what I focused on. We also know that there is an altered dopamine signaling. I didn't show you. We did cognitive training, half an hour, 5 days a week, 5 weeks. We could improve their cognitive functioning. We could reduce the clinical symptoms. We could see that there was a change in brain activity. We could also see that there were molecular changes in the dopamine system. So of course, these are separate, but again, it suggests that you here have a system that you actually start with the molecular changes, the genetic changes, and then you have this. So that was the end of the first part, the basic science. So I think we can take a 5-10 minutes break, and then I will go now more into the clinical and the epidemiology for the next one before we start the group works. Before that, do you have questions for this one? I can go back to this one, which is kind of the summary of it all. Yep.
You just say that this is basic research.
What you say?
I think that you said that this is basic research because the cognitive training is not clinical research.
I say it's basic research because?
The last thing that you have said is that these both studies were basic research.
Yes.
But I think that cognitive training is not clinical research.
If it's clinical research?
Yeah.
Yeah, no, but I mean, there are no strong, abrupt, and a big gap in between. But if you saw my title, I wrote proof of principles. Mm-hmm. So this is the first time anyone are proving that this principle is right. And it's actually done in experiments. We are not using mice, but we are using humans. We are using students. We are also using children with ADHD. But we are showing for the first time that this principle is there. And now after that, if you remember, there have been, I wouldn't say millions, and I wouldn't say thousands, but I can tell you hundreds of clinical studies where they have used different variation of this. And these have also been implemented. So this method is now used, I wouldn't say in every country, but on all continents, they are using different variations. And that has been clinical research, a lot of clinical research. But of course, we did intervention study where we controlled before and after. We have never done a randomized control study where we shown. So somehow there is a transition into clinical research. So this question then is, how many children with developmental disability and cerebral palsy are living in the world? Anyone have a qualified guess? So if you go to different UNICEF, for example, or WHO, you will find an enormous span from maybe there are 50 million children up to 250. So when you start now digging into it, what are these numbers based on? So you search here, and I started with one on cerebral palsy. So this is one of the last systematic reviews and meta-analysis of the prevalence, and it was published 2013, so it's kind of 5 years old, but I haven't seen anyone after that actually. So here you see they did then a systematic review on all papers published before 2013 where they had the prevalence of cerebral palsy. That's what they did. And they had two. A is ordinary, B is if they were preterm born children. And what they find then, now I have to do this, it's too modern for me. You see, this is actually the mean, which is 2.11. But then you can see that the different, Here is one outlier, and there are two others. But otherwise, if you look on all these studies, they show a prevalence just about 2 per 1,000. So let's focus on that one. You have then the other ones on the B side. But when you look on from which countries are these studies coming? So there were 48 who actually met the inclusion criteria, 48. Of them, one was from China, one was from Turkey, one from Kenya, and the rest was for high-income countries. So how many children are living in low and middle-income countries? Or you could just say percentage. So of the population of the world, how many children are living in high-income and how many in low and middle-income countries? Anybody knows? Somebody knows? More than 80% are living in low and middle income. So do you think this is a representative figure? Would we expect that the prevalence is similar in low-income countries as it is in high-income countries? Would you expect that? Remember my slide now with all the environmental risk factors. Would you expect it's the same, or would you expect it's different? So we could simply say nobody actually knows how common it is with cerebral palsy in the world. We know for less than 20%, we know, but for the rest, we don't know. So that's a big, I would say that's a big question to answer. If we look generally, have we better knowledge about the wide spectrum of disorders, including then cognitive disorders, sensory, that's hearing and seeing, behavioral, that's autism, ADHD, obsessive disorders, communication disorders, learning disorders. Yeah, so this is also a bit old, but there are actually none better. This was from 2007. So you could see then, and now it's from low and middle income countries only. Low and middle income countries only. So you see sorry, this is so advanced, so I have to reprogram myself. There we are. So you see, these are the countries, and then you see these are the screening tools. So it's mainly surveys and questionnaires to parents. And here you see the prevalence. This one is 0.4 in Bahrain. So that means that 4 in 1,000 children have a developmental disability. And then if you go up, here we have in India, there you have one 12.7. And if we go on, you see it's very well. So actually, and this author also claimed that all these studies use different methods, different tools, everything different. You cannot really compare. So it's and this is a latest. So that means really they are not much better for general. This is actually one of the few studies, and this is from the DCD in Atlanta in US, where they have the epidemiology of the United States, based on surveys. And if you look to the right, you can see the prevalence 2008. And you can see first, any developmental, it's about 15%. And then you can see the different autism, for example, 0.74, ADHD, 757, and so on. So this is actually one of the few where you have an entire mapping of all. Otherwise, the studies are more like the CP study, where you are only looking on one disorder and do not take into consideration the comorbidity. And then this study was published last year in August. And you see, Lancet Global Health. Now, you remember me telling you that it's important that you publish in high impact, you get citations, because otherwise nobody will read your your study. This Lancet Global is the #1 in global health. So they published this one, Developmental Disabilities Among Children Younger than 5 years in 195 countries and territories, a systematic of the global burden of disease, developmental disease. So then we thought, my God, now we will get the numbers, okay? So remember now, 15% in United States. So here the global estimate is to the right, 2016, 8,367, that is about 8.4. So they do the global estimate of children with disability to be 8.4 per 100. You follow that? Okay. A bit low maybe. You would have expected that it's more common in these countries, but maybe. But then you continue, and there you can see now on the top you have ADHD, you have autism, epilepsy, intellectual disability, hearing loss, vision loss. Total 8.4. Focus on this. This is a trend, but focus on this 2016. Okay, what is the blue one? Look here, 4%. It's actually half. Half of all children with disability, they are blue. What is that? That's vision loss. Okay. And then number 2 then, it's also quite much. It's more than 2, it's almost 3, the lilac here. That's hearing loss. So if you now add vision for hearing, let's say 2.5, that's 6.5 of 8.4. That means only 2% for the rest of the developmental disabilities. Do you remember this one? How much was hearing here? Okay, let's see, hearing. Where do we have hearing? Can you find hearing here? You're there, blind, unable to see at all. 0.13 compared to 4. Is that a difference or? It's a significance. The 4 compared to 0.13. Okay, where do we have hearing here? Hearing. I can't see hearing. But that means here they present 15% children with developmental disability. And hearing and vision is not 1% even. So, do you think this is, what do you think about this? Is this believable? Would you trust this if you see it? So this is what you have to do. I mean, you cannot just read and take in everything. You need to start asking questions. You have that, you have this. Can this really be true? Even if it's published in the #1 international journal in that field. Can you believe it? And then look at this. ADHD, 0.1%. Autism, 0.7%. And I have done quite a lot on both of these, so therefore I said, this is ridiculous. This is ridiculous. So let's skip the vision part because I already done that. Cerebral palsy, how common was it with cerebral palsy? There were no cerebral palsy. It was not even counted. Cerebral palsy is probably the developmental disability with the highest burden of disability in the world, probably. But it's not even included here. It was included, when I started to make fuss, they eventually told me that it was part of intellectual disability. Okay? So look at attention deficit, ADHD. You see in Australia, they say that it's more than 3%. You can see the red one. In Sweden, where we know it's 5%, it's reported as 1.4, 1.6 here. In US, where we know it's even more common, it's yellow, it's kind of 2%. And so if we then look, this is a global report. Here you have similar as I showed for cerebral palsy. This is now for ADHD. And they are now, if we see the geographic location, you see this is Africa, this is 5%. 5 is about the average. But you have several studies from Africa, and you see if anything is more in Africa. You have in Middle East, you have in Oceania, you have in South America, also almost most, Asia, North America, Europe, and so on. It seems to be around 5%. So in this one, they say ADHD worldwide pool prevalence is 5.29, and then they say that the large variability is probably depending on it. And as you remember, I told you that 75% of ADHD is genetic, 25% is risk factors. There are more risk factors in low resource. So if anything, you would expect higher prevalence, unless they are dying off before. Otherwise, you would expect that. So that this was not, so actually what we actually did here together with Diane Damiani, we actually write a commentary here that poor data produce poor models. Children with development deserve better. When we were actually telling them that, okay, you can have all these fancy models, but if you put in, we didn't say garbage, you get garbage out, but that's what we really mean. So I think this really tells you now that here we have an enormous gap. And recently you had this one, it was published 2015, where they looked upon different data for developmental disability. And you see here again, the enormous variation from 1.5 in China to 13% in Armenia. And you had some countries with very low, between 5, others with high, 10, up to 48%. So we could say here, there is an enormous range. There are some higher and some are lower, but there is an enormous gap here. So if you really want to do something on a big issue, this is what you could do. So these are several of the reasons why there are so big variations. So I would say from high-income countries, we have rather stringent, robust, trustable data about single, like cerebral palsy, like ADHD. We know just it. But as you know, it's more common to have 2 functional limitations. If you have 1, it's more common that you have 1 or 2 than that you only have this one. So therefore, you don't get the entire spectrum. And there are a few community-based broad-spectrum disability studies, and there are different skills. So how do you calculate prevalence then? Yeah, it's very simple. You just divide the number who have this condition with the population. Very easy, but the problem is, how do you get it? How do you get the population in a country where you have no registers, where you don't know how many, for example in Uganda, you don't know even how many people are living in Uganda. You don't know how many children are born every year in Uganda. And I think this is the case in many of these countries. And then out there, how can you get these children? How do you find them? What instruments? How can you screen? So this is really a very... So here is then epidemiology of cerebral palsy according to the SCPA, you know, the European surveillance CP network. A third are unable to walk. A third have severe intellectual impairments. 10% have a severe visual impairment. So what is the prevalence in a low and middle income country? So we actually now took the advantage of what is called a HDSS, Health and Demographic Surveillance Site, in Africa, in Uganda, and in the district. You see the green district here. Here you have the green. So within this little green district, this is Lake Victoria, here is Kenya. It's a rural area. 80% of families are subsistence farmers. There are 80,000 persons living in this area. Every year, the HDSS make a census. They find out how many newborn, children are there? How many have died? How many have moved out? How many have moved in? So what does that give us? The denominator. We have a population. Because otherwise it's extremely difficult. So we know every child living in this area. Okay, that was number one. How do we now find children with cerebral palsy? So what we did was a rather new way. We had what's called a 3-stage screening, where we first used the infrastructure of the HDSS field workers. So when they were doing their annual survey, we gave them 2 screening questions. Okay, how do you formulate screening questions for cerebral palsy? You cannot just write, do you have any child in this household who have cerebral palsy? Nobody would understand. So in this case, we actually now we are coming to the qualitative studies here. So how can we now know how to do a question? First of all, stigma. To have a child with a disability in Uganda is very stigmatizing. You hide them away. You don't want neighbors or others. So we had to take that into account. And what were the names they were using for children with cerebral palsy? So we had then focus groups with parents, with villagers, with different community leaders. We had interviews with traditional, both Christian and also traditional. So actually there were a lot of open interviews where we tried to find out what is your attitude to these children, what do you call them, what is the reason they have got it. This is a lecture by itself. I just tell you, it's extremely important when you formulate a screening question that you know the context where you're going to do it. So that was the first. The second one was that we now taught a mobile team, so they could go to the family, they could check the child, so we had some simple examination which a lay person can do. If they still were positive, they were coming into the hospital. So the assessment then was the screening questions. Now diagnosis, how do you set the diagnosis? So this is now how to identify the denominator. I told you, we had that already. How to find children with suspect CP and how to formulate the questions. And this is actually the paper not yet published where we have this qualitative study where we did that. Now, in order to compare cerebral palsy in Uganda with other places, we need to have the same definition. And if you go out in these countries, in the old literature, there are many, many different classification systems. And this is extremely important. Let me give you an example. I was in Russia quite a lot, maybe 10, 15 years ago, and they had actually twice as much cerebral palsy as we have in the rest of the world. And I thought that, I tried to understand why, how can that be that it's more common with cerebral palsy? But then when I were there meeting these children, I thought, but this is not cerebral palsy, this is a microcephaly, this is a neuromuscular disorder, this is poliomyelitis. So then I tried to understand how can you put, but they said, you know, if we give them CP, they get a lot of benefits. So going back, it found out they had a Professor Semanova, a hero of the Soviet Union, sitting in the high up in the party. She was very much devoted to children with cerebral palsy, worked a lot with them. She managed to get a proposal through the Soviet Union government that if you have a child with cerebral palsy, you should have special benefits, you should have services, you should have assistive devices. So what happened then when you have, as a doctor, rehab doctor, you're sitting there and giving the diagnosis. Of course, you know, if I put CP, they will get a lot of benefits. If I don't, they won't. So I don't know if this is the true reason, but it's plausible, and I think so. So anyway, you have to be very careful now. What is it? And here, you know, we have a classification system since 2000, beginning of 2000. This is kind of a key publication for cerebral palsy, which always you should use. And this is a European network then, the surveillance. And here you have a flow diagram, how exactly you set the diagnosis. So by using this, if you want to do this kind of studies, you can now compare with others using the same. But if others have used other older systems, it's very, very difficult to compare actually. Further on, you have, this is now how you divide them in different clinical types, in the spastic, in the ataxic, and in the dyskinetic. And then very important in the new system is now, and you heard quite a lot about the ICF, it's not the health condition per se, that's not so important. The important thing is the functional limitation. So here you can measure now the functional level of motor abilities. You have the gross motor classification, you have a manual assessment classification, communication, and eating and drinking. So if you use the diagnostic pattern and these classifications, then you have defined your sample in a very, very good way. So this is what we did. So we did a survey by the field workers asking 2 screening questions, which we had then put a lot of emphasis to get as sensitive and specific as possible. We had then trained mobile team visiting 420. So 420 of, are not, as you see, because we had, my God, you had, here you see, there were 442 were positive screening, 22 never showed up, so we had 420 which was assessed then. And then if we continue, we had of these 420 assessed, you see here, 233 were positive and were sent to the clinical specialist team. 7 did never turn up, so there is kind of a loss all the time. And of these 226, 140 were negative, but 86, they were then confirmed using the SPCE. Now, we heard that there were actually children out in the village who had not been included. So we did what's called a triangulation. So we did another way. So we went out to each village. We found out key informant health workers and asked, do you have this child and this child in your village? Do you have any other child with a similar type of problems? And in that way, we got 45 more children and 11 of them had. So that means we could now calculate the prevalence after triangulation worth 3.1. That's about 50% more than, and these data are very, very, very short, clear cut. Now, when we looked here, you see to the left, can you see that there are actually 16 per 2 age group. This is 2 to 4, 4 to 6, 6 to 8. But then suddenly there are only 8. Can you see that? And if we calculate the prevalence is actually 4, and then it drops to 2.0. So how can we explain that? How can you explain that? Huh? Who knows? Because if you look here, the blue one, there you are. You see that these are those with GMFCS 4 to 5. So there are several of them when they're young, and then they disappear. And we know actually from literature over in high-income countries that there is an increased mortality of children with cerebral palsy in 4 and 5. So this is our also suspicion. It's our suspicion. But that also means that the prevalence is actually higher during the first years. And we also think that this decline is due to increased mortality. This one we can skip. So anyway, this was the first population-based study from a low and middle income country. Last year it was coming a study from Bangladesh, which was the second. But interestingly, they had used another method. They had used what's called key informants. So that means they do not really know how many children are living in the area and people moving out and in. And they were only going and teaching, training community health workers to say, this, do you have any child with this kind of thing? Then you should go to this clinic and we will examine you. So it's a community-based, but it's not really clear how big the population is. So anyway, when we now compared our results with their results, we found a lot of similarities. So I think this is really good. But what we also found was there were some differences. And you should help me to explain why are these differences. So, if you look here, you have the GMF-CS level at the top. 1 to 2, 3, 4 to 5. They have the max levels. Intellectual disability, which is quite common, as you see, it's around 50% in Uganda. Similarly, this is from the Sweden and Australian cohorts. You see, it's quite common with intellectual disability. Seizures are very common. Speech problems, very common. Hearing impairment, not so common actually. And vision impairment, also not so extremely common. But now when we compare, you see in Bangladesh, they had actually much more severe cases. You see, they have 4 to 5, they have 47%. We only had 10% of them in the older age group, 36, and here you see the decline. But watch here, in high-income countries, there are 36. So they seems to have much more severe. And why do they have more severe in Bangladesh? So that's one question. The other question is, if you look on intellectual disability, they have actually much less than in Uganda. And it's about the same as in high-income countries. Seizures, they have much less than in Uganda, and also in high-income countries. And speech, they have then about the same. Let's see, yeah. But anyway, intellectual disability and seizures, they are much more common in severe cases. So here can you see that there is a paradox here. So what is a paradox if you look here now? So what I just told you is, in Bangladesh, there are more severe 4 to 5 than in both Uganda and high-income countries. But there are less associated impairments. So what would be your, is this possible? So you can read that paper because there we make something. So I think the design, we used a 3-stage survey where we actually tested them, and by every age we could find this out. They had this key informant methodology where you do not really know if you get all children, and also the denominator is not really clear. It's also important to use the right classification system. I think they did. But then the assessment of function and of associated impairments are very much important. And then that you have trained the staff. So our suggestion, but they have not responded to this, is that these differences are due to methodology, not to true things. We think the key informant system is not picking up the mild cases, mainly the one. We do not think they have used stringent enough to look at associated. But it's up to them now to respond to this. But I mean, it also tells you that you have to be very, very, very rigorous when you do these kind of studies. Okay. I just can give you some more, because doing this gave us a lot of new unexpected data. So we know that in high-income countries, preterm birth is a major cause or major related to. So if you are born before gestational week 32, you have a risk of 20% to get cerebral palsy. Wrong. If you are born before gestation of week 32, 6%, you have a 6% chance. But since that is so prevalent, you actually contribute with 20% of children who get cerebral palsy. If you are born between 32 and 36, the risk is 1 per 100, it's 1%. But since this is a large proportion, they contribute to 30% of children. So that means that about 40 to 50% of children in high-income countries, they are born preterm. And you see in the full term, it's one per 1,000 then. So now look in Uganda. So how many preterm did we have? We had only 2%. So that's huge difference. In high-income countries, 40 to 50. In Uganda, it's only 2. So why is that? What? They don't survive. So they have not developed. And now it's interesting. So they have big campaigns now for stillbirth, it's called, to save these children. And what we say then, okay, if you do that, you have to think about the cost. So if you do it, you have to do it, not just save the life, save the brain at the same time. Post-neonatal, do you know what that means? That means if you get the event causing your brain damage after 1 month of age, 28 days, then it's post-neonatal. So this happened in Australia, in their register, about 5 to 6% of children with cerebral palsy in Europe probably have a post-neonatal origin. And look in Uganda, there were actually 24. And in addition, in our screening, we found 4 children who had their damage after 24 months. They had a CP phenotype, but they had the event after 2 years. And then we call them acquired brain injury instead. So why is that? Then you should know this is an endemic area for malaria. So what these parents now, and this is now qualitative research, we had interviews where they should explain different things. And one was that, my child was doing perfect until she was 9 months of age. Then she got fever, she got seizures. These seizures were going on for 2 weeks. And after that, she has now she has a unilateral then cerebral palsy. So, as you see, 1/4 of the children then in this area had a post-neonatal, probably from a meningitis, encephalitis, or malaria encephalitis. And this is, of course, preventable, like many of them. Then we also now come into the next part, the health services. So what kind of services are these children getting? So you see, of the 37 children who could not walk, 2 had wheelchairs, 1 had a walker, 40 children were carried, and 19 they could roll and so on. So that means there were new assistive devices. If you had had the same sample in Europe, hopefully more or less everybody should have had some support. When we asked them why didn't they get support, you see that they had given up. They had no money, they had no knowledge, and they had lost hope because nobody could help them with their child. So that means there were very little services available. And now we come to the ICF participation. And one proxy could be how many of these children were in school. And you see that if you had a GMCS 1, 40% were in school. In 2, 30%. And 3, maybe 5, and 4 and 5, none were in school. and actually seizures and the other associated impairments. So you could see in Europe, I would guess all children somehow will get schooling, but that was really not part of it. And this is now something leading to the next, and then I only have 50 minutes to go here. So now we actually observed, when we were checking these children, that they were actually not developing supported mobility, and they were rather inactive when they were doing things in their hygiene or helping in the household. So what we did is we did the PEDI score, and again, can you use, do you all know what PEDI is? It's an ability. What can you do? Can you use the elevator? Can you use the toilet? Can you put on your winter jacket yourself? Can you jump into the car yourself? Do you think you can use that kind of questions in Uganda? Of course not. So first we had now to transform the PEDI, which is used everywhere, to Uganda. So we have now a Uganda version of pediatric disability index. So now we could see, here you can see the mobility, and that, my God, that one I don't really learn. So here is the mobility index, this is age, and this is the mile, GMFCS. The black are Ugandan children with cerebral palsy. The white, they are from a cohort in Sweden. Can you see a difference? Yes, it's a statistical difference. Both cohorts increase in mobility with age, but the Swedish cohort increase faster. The next one is now the DMFC3. The white is a Swedish cohort, and you can see with age, mobility increase. In Uganda, it does not. And similarly, but of course in 4 and 5, also there is very, very little increase in mobility in the Swedish cohort, and absolutely none in Uganda. Similarly, when we look on MAX, and I think you will learn much more about MAX tomorrow with ability, you see it's similarly, the white increase steeper, faster than the black, the Swedish CP children with self-care ability. In MAX 3, it's very clear, you do not have a development in the Ugandan children, but you have in Sweden, and similarly here in. So that means, so you know the classification, it's supposed to be stable over time. That's the whole idea, because you should be able to see the development. And if you have a GMFCS set, you should have a very, very slight, while in one you should have. But this actually tells us that this does not really apply for the Ugandan children. They have less developmental mobility, less development of self-care skills, in particularly in the middle. So this opens up now, what can we do for these children? So this is a summary of it, but in order now to 10 minutes more, I will go to the next. So what can we do? Can we, by applying services, by intervention, can we provide better development for these children? Let's see now. Here we are. So maybe I should open up for question first. I think you will have very little lunch time here because I'm talking too much. Please, questions.
Use of wheelchairs in low-income countries is very difficult because of the surface. Have you on this or not yet?
So I think this is an enormous area, and I think there are many, many reasons. And one is, if you're living in a clay cottage with dirt ground, dirt roads, where it's raining, I mean, how can you use it? But you know, one thing is, by sitting up in a wheelchair, you can start to participate in the family. If you're lying down in the corner, or even worse is you are lying out with the chickens, you cannot participate. So that's part of it. Another thing is who provides these wheelchairs. And then there are a lot of non-governmental organizations. They have bought 40 wheelchairs, they go out to a village, they find children, they give them out, and then they disappear. And what's happening with them? We're actually doing now a study on that, but we don't think that helps very much. So assistive devices has to be incorporated in a more holistic way. And this is what I will try to show you. So it's a very good question. Yep.
I think you make a very good point, and I think that if the assistive devices are developed within the country...
Can you hear at the back?
Can you hear? If you develop the assistive devices in the country, they're more likely to be appropriate. So I've done some work with the Indian Institute for Cerebral Palsy. They have a lot of children who need high-tech communication aids, but the ones from high-income countries are just so expensive, it's not realistic. So they've worked with an Indian tech company, and they've produced a voice output communication aid, which is within a reasonable cost for at least a middle-income family within India, and has appropriate language built into the system. So by working with indigenous organisations, I think you get much more appropriate technology.
Absolutely. I completely support you in that. More questions? So then I will go through the last part here, and maybe I will take 10 minutes of your coffee break, but we will see. Maybe I can quick it up. Okay, so this is the last part of my presentation then. So now you heard one of the reasons why we need to develop an intervention for children with cerebral in low resource settings. That's quite clear. And how can we evaluate the effect? Because you know what we saw is that the functional development seemed to be clear. We also saw that there are no services. They had no assistive devices. Parents have given up. Probably there were a higher mortality rate. So there is obviously a lot of things that could be done. The first question is, are there any evidence-based intervention that can be used in this low resource setting? And now you have to go out, do your literature search. Evidence-based, that means there are a lot out on the internet. But if you also set, we want this intervention to be tested under a research condition and have been shown to be effective, there are none. There are many different, and when you are out like these people, oh, do you know that person? They are doing a fantastic job at that NGO in Kenya, or these are doing something fantastic in south of Zimbabwe, and so on. And there are many people who are really doing a lot of contribution and helping, of course, these. But if you want to scale it up and get kind of on a more international, I think this is the way you have to do it. It takes time, it's tedious, but you have really to prove that what you have done, because then others can use it and they can argue that this can be used. So how do we do it now? And this is now general, not just for this one. So if you want to find out evidence-based practice, you see you have 5 steps. You have ask a clinical question, and that we did. Are there any intervention for cerebral palsy which can be used in low resource settings? Then you have to acquire the best evidence. And how do you do that? I will show you. How do you appraise the evidence? Are they really valid? Are they evidence-based? How do you apply them? And how do you assess that you have? So this is in a way similar to the start I had. You will have these one because these are fundamental steps when you want to introduce something new. There is also something called PICO. So PICO stands for population, P, intervention, I, comparison, C, outcome. So this is a general thing which can really help you. And this one I took actually from Kate Benfer in Australia, who have been lecturing for the IICD. So her question was actually, is it effective to give nerve growth factor on motor development for children with cerebral palsy compared to standard care? So that's a rather limited. If I go out with my big one, it will be much more difficult. This is kind of a... So this is actually what you should do when you now test your own project. You should really go in and find out what has been done before, because that we are reinventing wheels everywhere many times. That does not move the whole area forward. It might be good for your local community where you are, but if you want to really reach a higher impact, this is what you have to do. And then you have now to acquire best evidence, and there are, as you know, many different databases, the PubMed and Medline and many others. There are also guidelines. The NICE, for example, are very good. That's from the UK, and there are many other guidelines you can go through. So when you now acquire, you have to do this research, this search in the literature. So you see up, you have the PICO, you remember, the population. So it's cerebral palsy and it's infant. You have the interaction, that was a nerve growth factor. You had the comparison, that's standard care. And you have the outcome, and it's some kind of motor outcome. So this is the way you should construct your search. Then if you look down, you could see you have a list of synonyms for each column. So in addition to cerebral palsy, you say disability, neuromotor impairment, and instead, not just having infant, you have child, you have pediatric, both American and British spelling, and so on. And then you combine when you search with OR. So that means when you now search, you will get all papers with cerebral palsy, disability, and neuromotor impairment. And then you do the column S1, 2, 4, 5, and then you do the end. So that means you will now get all papers published with cerebral palsy or infant on those columns, and NDF and that columns, and standard care, and motor outcome. So that means now you will get everything on the database, and you will probably, I don't know how many she got here, but sometimes you get several hundreds when you do it like this. So now you have to appraise the evidence. How certain can you be that the stated evidence is true and that it has benefits and harms then? So then there are quality of methods which minimize bias with study design, relevance of the evidence, how appropriate is the outcome measure for the problem you want to do, and the size effect, how strong is the evidence. And this one, I guess you all know. So this is, it used to be called a SACET, but it's more. So this is the highest evidence is then if you have randomized controlled trials, and in particular if you have a systematic review of randomized trials. That's the highest level of evidence. So if you are lucky enough and find a couple of systematic reviews on RCTs, then you are home. Then you could say, here we have strong evidence. Then you have the cohort studies, and if you have a systematic reviews, it's of course better, but it's not the highest level, and so on. You have case control studies, then you have case series, and you have expert opinions. But you should at least be up to 4 or 5 to say that you have some reasonable evidence. If you are looking on guidelines, there is something called AGREE, appraisal of guidelines for research and evaluation. I have done this now for the WHO, who are now developing a package of rehabilitation guidelines for cerebral palsy, for example, and for autism spectrum. And then you see you have 23 key parameters you have to look at. And you see there are 6 different domains where you very, very carefully go through that everything is covered. And if they have not really covered all these with a high, they are not really counted. So that means if you want now to be having an impact, you have to do highest level of evidence, and you have, when you do these guidelines, you have to actually follow this in advance. So this is actually now 2013, it was published by the Australian group, where they used this, what you call grade of evidence, where you look on the quality of the study, the recommendation, if it's strong or weak. And they see, in this case, they looked upon intervention for cerebral palsy, and they had 33,000 articles to start with, as you see. And they watered it down in the first step to 231, 171 by having inclusion-exclusion criteria. And eventually, as you see, they had 165 articles that met. But you could see from 33, it was actually only very few proportion that could be used. So please, when you do your studies, see that you are in that category and not in those which cannot really be used for this kind of translational work. And this is kind of a very famous picture where the green ones in the grade system show, do this, strong evidence, strong recommendations. The red one, interestingly, showing new evidence. Don't do this. There are many, many studies, don't do it. And there you have, for example, the BORBAT NDT, you have the sensory integration, and you had the hyperbar oxygen treatment. So there you have treatments that has been tested, which has been shown, don't do it. So when I'm out traveling, do you think I see any of these treatments anymore? Very, very common. So it takes such a long time. Now, we need much more of this implementation research. How do we now use the new science and develop new treatments? So then we should now assess performance, and there is another system called SMART, where you should then use specific, measurable, attainable, relevant, and timely. You will have this as a PDF. I will not go in the detail, but there are systems now when you start implementing it. So ensure by using valid and reliable outcome measures, goal-setting goals. If you don't have good methods, don't do the study, because it will not then be able to be used by other people. Now we should apply the evidence. Then, of course, the best available research evidence is just one part. You have now to see what is the population you want to use it for, what are the resources you have, what is the environment. So of course now, we have this evidence, but how do I apply that in Uganda, where 80% are living under poverty level? So you cannot just take the best evidence and go there. You have really to do something different. I guess for you, there might be other barriers, other things which kind of define what you can do, what you cannot do. But the system. Then now, before starting your study, go and see where am I going to publish this. And then, you know, for example, DMCN, Developmental Medicine Child Neurology, they use this Equator platform. So if you look to the right, reporting guidelines for main study, then you can see that you have, if you're making a randomized control trial, you should use the consort protocol. And it tells you exactly what you should do. Because then when you submit your manuscript, you have to check in all of these things. And if you not from the beginning have done it, you cannot recover. So before even starting your study, go in and see. If you are then doing an observational study, you should use another protocol called STROBE. If you do systematic reviews, there is one PRISMA, and so on. There are others, but you cannot just start and then do this at the end. When you have done your research questions, when you have found out your design and method, please go in and check that you haven't forgotten that you are. This is extremely important, actually. So this is just an example, and you see there are millions of things you have to think about, but you have them so you can check out one after the other. So now we come into the intervention. My God, I'm too slow here. I will just give you now an example how we have used this. So we now want to see if we now implement an intervention for these children, how can we do that? So then, of course, we need to have a randomized control trial, because that's number one, you remember. So we do 2 arms. We have 1 treatment control group and 1 control group. We randomize, we use placebo, or we do not. It should be blind, we should stratify, and there should be enough children. This is a ground thing you have to do. So in our case, we will use 4 components. One is a care-to-care program, and that's based on Gillian Salyone, working in South Africa for 30 years. She's a physiotherapist. She has developed over the years a care-to-care program where you really teach parents, because there are no doctors, no therapists, no health workers, nobody out there know anything about cerebral palsy. So she has to build up a parent or a care-to-care network. We are using goal setting, because goal setting were one of the big green balloons, if you remember from the review. We are using assistive devices because we definitely found out that there are no devices. And we use the 4th, communication and advocacy, because in these qualitative studies we found out the stigma is completely blocking everything. So in order to get these children into the early child development, into school and so on, you need to break down the attitudes in the society. We have to strengthen the child and the family. So this is the 4 components we are doing. We are doing this simple, but then how do we randomize? Because if you have communication and advocacy, that means the whole village. So what we do is we take part villages being controlled and the other the experiment group. So it will be on the village level. And how do we do a control group? Yeah, then we can only do 40 children per round. So that means for the first year we will now do 40 children in part of these villages, and 40 will be on the waiting list, and they will have the treatment the second year. Of course, we also have to stratify with age and with GMFC level. We cannot only have 1, 2 in one group and 4, 5 in the others. There are no 4, 5, by the way, but 3. So, and what outcome measures? And what do we want to measure? And then you talked a lot yesterday. You could say body function and body structure is kind of important. But for these children and for these families, the most important thing is actually activity. What can they do? Can they take part in the household? Can they be with their and it's also the participation. Can we increase school attendance, for example? So that is very, very important now that we have that. So what we have come out with is actually we measure at baseline first year, second year. We have several outcome measures for functional ability. We have parent skill and well-being. And we have the effect of communication and advocacy. So these are a battery. And of course we have to train the staff very, very carefully. Okay, that was the end of that story. And now we are about 15 minutes late. But we actually started 5 minutes late, so it's only 10. So the idea now is the following. You will have coffee. And we have the time, it's not so bad actually. So why don't we do like this? We have coffee until 11, 20 minutes. We come back, then you will have 4 of your examples. They will be presented. Then we will have a short group. You will be in 4 groups, each having 1 mandatory and 1 optional. And then we meet again and we have a discussion about these projects. And try then to see if you can remind some of what I have tried to tell you about the stringency and so on. Before that, do we have any questions now here, or should we wait until coffee and have general questions? I think we take coffee and then we go on. Good?